Today I'm going to introduce to you my research on enabling scientific research with language agents. So before we dive into building scientific agents, let's first look at what us human scientists do. So I asked Claude here to give me a good example. So the first task we want to do is we want to label all the daily tasks that cannot be done by the scientist alone. We can see here that in the early morning, the scientists will want to review some literature and preprints, its interaction with Google Scholar and Semantic Scholar environments. And then, no matter it's a computer scientist or a web lab scientist, the scientist will always want to interact with the laboratory environment to verify his ideas. And finally, science is all about communication, and the scientists want to interact with other humans. So human is another environment to interact with. So now we do a thought experiment. What if we take all this away and we lock an AI scientist in the box? What will happen? Supposedly, we give the model a research question to solve. For example, what will happen if there's an EGFR mutation in lung cancer patient, and what will happen to the immunotherapy response? So this is a novel question for the AI scientist, and because it has no knowledge based on this question, it can only make an educated guess, which will not be very good for us. So what will happen if we instead attach it to a research environment? So the large language model agent could start a workflow. We can see here that first it can search about hypotheses. A simple hypothesis will be immunotherapy response is very related to some genes, and we'll look at what kind of genes in fact immunotherapy response in Semantic Scholar. And after searching for a bit, it will find that PDL1 gene expression is very related, and then it will try to plan an experiment. So because it's an EGFR mutant patient, so we want to measure what kind of co-expression exists within EGFR and PDL1 gene. And in the end, it can get some experimental results. And as we can see in this figure, this is a result from microscopy. We can see that that the patients with EGFR mutant has higher PDL1 response. And in the end, the scientists can form new hypotheses based on experimental results. This is a loop of an agent. So we can see here that with a large language model agent and an attachment to a research environment, agents are much more powerful in doing autonomous scientific discovery. So how can we build this kind of systems? The first question is, do we have strong enough agents to perform research level tasks? The second question is, do we have easy to use tools and scientific environments for agents? In this talk, I will be presenting about two parts. The first part, I will talk about agent for science. So we first look at what kind of systems we want to build for agents to work in scientific environments. And for the second part, I will talk about how to build strong enough agents to solve research level tasks. I will focus on one specific agentic ability, which is improving the long horizon planning abilities of large language model agents. So let's get started. So we'll be focusing on a specific kind of research environment, which is a database environment. Database is very essential, especially for biology, because it's a very knowledge-intensive task. I'll be introducing two examples. The first example is how to use a protein database, and the other example will be a disease pathway database, which is CAT. So what are the challenges of interacting with this kind of biological databases? We can see first that most of these biological databases are built long ago, maybe 10 or 20 years ago. So they are not very fast, and they're many slow tools that need modernizing. Also, a database is all about past discovered knowledge. How can we use the database to create new novel discoveries? I will talk about each part in one paper. So I'll first introduce our work on retrieve sequence augmentation for protein representation learning. So it's basically how to use a protein database to enhance understanding about proteins. So I'll first introduce some background about protein sequences. So proteins are chains of amino acids. Each amino acid is one of the basic 25 amino acids. So it can represent each amino acid as a token in like a sequence. The chain of amino acids forms some structures, and the structures define the function. So if we want to understand about the structure or function of protein sequence, we will look at its sequence. So how to build search tools for proteins? We are all very familiar with how to build search tools for text questions. So how to do it for proteins? So in UniProt database, they use a kind of search called blast search. First, they find all the related protein sequences. I'll talk about it more later. After finding all the related sequences, it will try to look at what are the common features of these sequences. We can see here by the entry name that this query protein sequence is very similar, over 90% similar to all kinds of insulin proteins. So this protein is very likely to be an insulin protein in one kind of organism. So how do we build this kind of blast search tool? They use evolution-inspired alignment to do this kind of search. So proteins exist in all kinds of organisms, and some organisms often share the same function. For example, we all need globulin protein to carry oxygen in blood cells. This kind of functional sites are very conserved because once there is a mutation in the conserved functional sites, there will be results in deadly diseases and will interfere with evolution. So during retrieval with multiple sequence alignment, we will first try to align the query protein with every protein inside the database. After that, we will try to see the maximum pattern between the query protein sequence with the search protein sequence. We will choose those proteins with maximum alignment quality. So by alignment quality, it means by the least number of substitution, which is token-wise mutation or gaps. Maybe there's no protein amino acid there, so I just delete those tokens. So how many number of substitution and gaps define the alignment quality between the two proteins. And after I found out all the alignment score of every search protein, I will choose the best ones. So this is what BLAST tool is basically doing. But there's a severe problem with this kind of MSA-based searches. The main problem is that the computation complexity is very high for this kind of methods. So it's originally an NP-complete problem, and you can solve it with dynamic programming. But recently, people has accelerated using HMM methods, and now the computation complexity is still polynomial to the sequence length. So if you have a very long protein sequence, it can take many minutes to run a single search. So now we take another thing, we take another thought on how to build very efficient protein search tools. In natural language search, we normally use a kind of method called dense retriever. So instead of doing token-wise alignment, which is very costly, we pre-train a dense retriever based on protein language model, and then we map all the protein sequence in the database to embeddings. After we have this kind of dense embeddings, we can use very fast nearest neighbor search techniques to retrieve the neighbor proteins for the query protein. And in this way, we have our new method, which is RSA. It is a substitute for the original MSA method to do protein search. And after using dense retrieval, we have sped up the retrieval speed to more than 300 times and has over 90% retrieval precision on PFAM database. So now we have a very efficient search tool. How can we use it? The first thing we can use is we can link it to a large language model to help the large language model understand about protein sequence. So we can see here that GPT-4 is originally not very well pre-trained on protein sequence. It can barely understand it. But after we link our RSA tool to the GPT-4, the large language model can first use the tool to search about the sequence and see that what kind of homologs the protein has. So by homolog, I mean the similar protein families. So we can see here that this protein is most related to the NMU protein family, and therefore it is very likely to be involved in bioprocesses such as cell population, proliferation, signaling, and programmed cell death. And after this kind of link to tool, we can see the GPT-4 plus RSA method has reached over 70% accuracy on gene ontology prediction. So RSA is not the only tool we can use to understand about protein. We only use it to understand our protein sequence, but there's so much knowledge we don't know with RSA tool. So we try to link RSA together with other tools to build a large language model agent. You can see here, we can first use RSA to search about the protein sequence. And we can see that this protein sequence is a ribosomal protein. And now we want to understand how this protein sequence can be used to target disease therapy. And I can search in PubMed database or UniProt database about the protein sequence. And in the end, I can know that this protein sequence is related to cancer therapy and neurodegenerative diseases based on this kind of information. So in this way, we can build a large language model agent to solve open-ended biomedical problems. So a second usage of MSA is actually to augment predictions. So we are very familiar with the AlphaFold 2. MSA transformer is a base model for the backbone of AlphaFold. So MSA transformer is basically taking in every MSA profile of every sequence, and then they try to perform a row-wise as well as column-wise attention to aggregate all the information of the MSA profile. But this process takes a lot of pre-processing due to the MSA technique. And we introduce another method, which is using our RSA retriever to retrieve all the related sequence. And without doing alignment, we just simply concatenate this protein sequence to the query sequence and then go through a transformer to do downstream prediction. Interestingly, MSA is not the best choice for retrieving protein sequence. Here is a map of ESM protein atlas, which is basically every protein humans have ever discovered. You can see here that there are many clusters which are protein families. But here is one sample of a protein that does not belong to any family. We'll call this kind of proteins orphan proteins. And predictions for this kind of orphan proteins often drop very significantly for models like AlphaFold. For example, without using MSA, AlphaFold 2 can drop by more than half in performance. So how can we mitigate this? Using dense retrieval, we can actually use a version of soft retrieval to find some faraway neighbors for those kind of orphan proteins. Despite they don't have some direct neighbors, we can still use some related families information, for example, like the structure or evolution information from other family proteins to help enhance the prediction for this kind of orphan proteins. Using our method, we have achieved over 10% improvement on average for protein understanding without using alignment during any process on a range of 6 tasks, including like structure prediction tasks and protein engineering tasks. So now I have introduced one notion of how to use a database efficiently. But what can we do if we want to do novel discoveries using databases? Here's a work that is very recent. So in this work, we look for one specific kind of scientific discovery, which is perturbations. So perturbations are one of the most important discovery things that we work on in biological system. So by perturbations are actually changes made to biological processes. If we want to understand how a drug actually works, we want to look at some effects of perturbation. For example, here in a taste transduction. Oh, sorry. Oh, maybe I can. I think I just accidentally.
Probably a blank button on the remote.
Yeah, I think so. Maybe I can share this again. Sorry about that. Yeah, I think I accidentally dropped out of the conference. So yeah, it's okay now. So we will go back to where we are at. So we want to understand the effects of perturbations on biological systems. So understanding of this kind of perturbations is same as understanding results for drug discovery or understanding biological processes. For example, here in a taste transduction pathway, we want to see what will happen if the M3 receptor is blocked. So originally, In this pathway, there is an acetylcholine that will activate the M3 receptor, which will lead to activated G protein, and which will go all the way into the COMH1 channel. And then the COMH1 channel will release ATP as well as neurotransmitters to activate the taste sensory fiber. So this is basically how our tongue processes taste information. But what will happen if the first receptor is blocked? We can definitely use experimental verifications to understand this kind of perturbations. But doing this kind of experimental verifications are often expensive. For instance, if our perturbation involves a gene, we should do a gene knockdown experiment, which is very, very costly using CRISPR method. And it's also, many scientists don't have access to the wet lab environment. So is there a faster substitute? Can we use a large language model as a faster verifier for understanding perturbations? So we first do a benchmark on how large language model understands this kind of perturbations. We divide the perturbations into 3 different categories. So we first look at a simple case, which is a natural prediction of the cause and effect in a biological process. So we first look at what's the effect of one component activation on one response. But when we try to perturb the system, so we add another intervention to the input variable. In this way, we no longer try to predict the original response, but the altered response. Another kind of perturbation is called interventions. So we don't directly interact on the input variable, but instead we try to change the middle process. So we add a change into a middle node, and we still want to see what's the final effect. And finally, we have a benchmark of various effects. So we have seen the simple one, which is just a single effect, what will happen if one thing is activated, and what will happen to another thing. But we also want to look at more complicated effects. For example, how one change in a component will affect the interaction of 2 other components. Also, what will happen if we have one change in component on the final organism. So we benchmark all these kind of things, trying to build a very good benchmark. So we try to do a rediscover experiment because we don't have the access to wet lab to create new perturbations. So we try to mine them through the literature. We first get access to PubMed, and then we try to get a pile of high quality papers. Then we try to filter all the papers with high quality pathways. And in the end, we have 6,000 pathway papers. And after we have the papers, we use the large language model to mine the perturbation contents as well as the type of perturbations, and we make it into a question and answer generation problem. This is the final benchmark result on this benchmark, and we can see here that the large language model originally has a lot of understanding about the natural biological process. We can see in the deep green bar and deep brown bar, it originally have more than 70% accuracy on this kind of task. But the performance quickly drops when it's challenged with perturb or intervene problems. So this shows that large language models itself has good biological knowledge, but their knowledge are not good enough to do verification for novel discoveries. So how a scientist will work when it's met with a new novel perturbation? So for example, we use the same example from just now. What will happen if the receptor is blocked in T cells? So for a human, he will first try to go up to a pathway database and see what kind of related information he can gain about this kind of question. And we can see here there is a subgraph in the taste transduction pathway that is very related to this question. And after that, we can have this kind of pathway graph. We will try to go through the process of the graph and see what is the cause and effects. So now we have blocked the M3 receptor. So there will no longer be activated G protein. So there will no longer be a surge of calcium ions. Without the calcium ions, the COMH1 channel will no longer be activated. So this will stop the injection of ATP as well as neurotransmitters. And in the end, the taste sensory fiber will not be activated. So the taste evolved responses will no longer be available. Inspired by how humans work with pathway database, we built a pathway graph agent to do the same thing. For a given question about a perturbation, we first try to find out related subgraph inside a large pathway database. After that, we will try to do a graph diffusion. So we first look at the most related nodes, and then we look at the neighbors of those related nodes. We'll go through the graph iteratively until the large language model believes that it has gained enough information about the process. In the end, the model can aggregate all the searched information and come up with a final understanding. Using this kind of pathway agent, we achieve almost 50% improvement on the perturbation and intervention set. So here is our research about agent for science, and now we try to move on to science of agent. If we have the good enough environments, it's not all we need. We still need a very good agent to do this kind of research level tasks. So are the large language model agents now powerful enough? So we will first look at a benchmark study we have done. In this study, we compare our evaluation of agent large language models against a commonly used benchmark called MMLU. So MMLU is mainly evaluating the common sense knowledge as well as basic reasoning of large language models, while agent board is more focused on agents. We can see here that there are many models that share almost the same metric on MMLU, but their performance as agents varies a lot. Another study using our benchmark for evaluation showed that doing some principal component analysis showed that agent board assigned largest importance to reasoning and planning tasks. So GSM-AK and Human Evo are both very challenging reasoning planning tasks, and we can see here that it's most similar to our benchmark in terms of ability needed. So we try to understand, is the reasoning and planning ability of large language models good enough? We do a long horizon analysis, so we let the agents interact with the environment for until 30 turns of interaction. And if we expect the agents to improve itself in every turn of interaction. However, we can see here that after 10 steps, most large language models, the performance saturates, and they no longer gain improvement after those steps. This is very problematic because in research, we often have agents that need to interact with the environment for more than 10 steps. And sometimes we even need the agents to interact with the environments until 100 steps. So how can we enable this? In this work, we introduce a theoretical inspired method to fix this problem. We first understand why is the long horizon challenge hard. We formulate planning as a trajectory optimization problem. So this formulation is commonly used in control theory. For trajectory, we want to find the best combination of actions that gets the closest to the final goal. So we can see here that there are T actions here with each action chosen from an action space. So it's a combinatorial optimization problem to find the final best combination of actions. But combinatorial optimization problems are notoriously hard when the length gets longer. So how can we solve this efficiently without making any mistakes? Here we propose a notion called non-myopic planner. So there are two kinds of planner. The first is one that is very short-sighted. So myopic means short-sighted, not very global optimal here. So a myopic planner only considers the next step and does not consider future consequences. But if we want an agent to be a good planner and can make a global optimal prediction and try to solve the trajectory optimization problem, this agent needs to take into account of future steps and consider the future consequences and then make a good prediction for the current step. So it's a large language model of regressive planning myopic. So the most commonly used methods for large language model, chain of thought, and React agents are mostly of regressive planning. So based on a length of history, the model is prompted with the history, and then it tries to generate the next consequent step. So we run a myopic test. So in a myopic test, we just try to figure out whether a trajectory is sampled taking account of future outcomes. We use a statistical test for this. So we find out that the myopic rate in math tasks for GSM AK and math, and we can see here that the myopic rate in running samples are significantly higher than correct ones. This shows that myopic generation is actually harming reasoning and planning. So how can we mitigate this? We try to figure it out by looking for a good algorithm in control theory, which is model predictive control.
Can I ask a question?
Yeah.
I'm trying to understand why, if you go back a slide, why in a correct trajectory you're getting my opticism. How if in the correct, how does the trajectory end up being correct if it's myopic?
So maybe like the model is myopic, but sometimes the large language model is trained very well. So despite it is not so global optimal, it can still make some good predictions.
So by correct, you mean it's not optimal necessarily.
Yes,
Okay,
but
thanks.
we just get good answers.
Thanks.
So I think myopic is defined with respect to the LM probability, right? So it's the trajectory is myopic if after you sample the trajectory, you realize that it's not actually the most likely output.
Got it.
Yeah.
Thanks.
But that still can be correct, even if it's not the most likely.
So now we try to solve this problem, and model predictive control is actually giving a good solution. So they try to solve the trajectory optimization we mentioned just now, but instead of optimizing all the steps as once, they try to break down into a consecutive optimization sets. For the first action, it tries to optimize the best action that will lead to global optimal decoding. And then it fixes this action and goes on to the next optimization of the next action. This process is repeated until the agent finally gets to the goal. So solving this problem solving model predictive control equation is actually hard because we want to take future optimal into account. So how do we do this? We propose a sampling mechanism. So instead of directly sampling based on history, we also use the model to perform a rollout of future steps. In doing the rollout, the model will try to generate not only the current action step, but also try to generate what will happen after this action to the environment, and what will go on, what kind of action we will also generate after that, and then we'll get a rollout trajectory. Then the model uses the probability score to evaluate the rollout trajectory, and we'll get a confidence score. So we not only combines, so we try to do a combination of both scores. The first is the original generation probability, and the other is the model confidence of the rollout of future steps. In this way, we can ask the large language model to be explicitly aware of the global future. So how to implement this? For the first solution, we introduce a test time scaling method. So we ask the large language model to do a rollout and to do a beam of rollouts. So in this way, we can know that some actions are better than others based on a confidence evaluation of the future rollouts. And after we got the scores of future rollouts, we will try to choose the best action for the current step based on the confidence score. So this is a test time scaling method because we can see there are two parameters involved. The first parameter is the size of the beam. So the more beams of future rollout sampled, the closer you are to estimating the large language model probability distribution. Also, we have a parameter which is the number of steps into the future. So the longer of the rollout into the future, the more accurately you will get to estimate the future consequences. So we try to make these two parameters find the best combination using the right budget, and in this way we'll get a test time scaling algorithm. So there are currently some very good test time scaling mechanism, including self-consistency and MCTS tree search. And interestingly, because model predicted control use a linear solution space instead of exponential one, sampling within that space is much more efficient than directly sampling from the trajectory optimization problem. In this way, we can get a little superior test time scaling method in terms of efficiency performance trade-off. Another method of implementation is to do a global local alignment reinforcement learning. As shown just now, we can now use the future to evaluate better trajectories for the current step. So now, instead of doing R1 kind of training, which directly uses a reward model or a verifier to find out whether the trajectory is good or not, we can now use future as a reward. So for each step, we try to generate a beam of trajectories, and then we try to evaluate the future of the trajectories based on the model confidence score. And now we would have a pairwise score, which is one is better than another, which shows us maybe one action is better than another at the current step. We do direct preference optimization training based on this kind of pairwise data. In this way, we don't need any verifiers and reward models, but we can still do stepwise reinforcement learning. The most interesting thing is we mentioned that non-myopic generation is basically trying to reach global optimal. So can this kind of ability be acquired through out-of-domain data and then transferred to other domains? To test our hypothesis, we use two general instruction tuning corpus, MACPy, as well as OpenHerms, to train our model on with our method. And then we try to do a testing on out-of-domain reasoning tasks, including math and logic tasks. And we can see here that with this kind of training on general data, we actually show a large improvement on other downstream reasoning tasks, even if we don't train on in-domain data. This shows that non-myopic generation is also transferable across different domains and can improve reasoning and planning. So for the last part, we will look again at the 10-turn challenge we mentioned just now. So have we now solved it? We can see here that using Llama 3.170 billion model as a base, we performed three different kinds of algorithms, including our introduced predictive decoding, as well as two baselines as a react. The baseline still saturates after 10 turns of interaction, while our proposed method can continuously reach a better result even after 20 steps. So now we look at long horizon planning in recent context because we know that recent models are becoming better and better in doing long horizon planning. Maybe 1 year ago, we're still stuck at a 10-turn problem with Llama models. And now, because of new advances, we can now reach 30 turns. We can solve more complicated problems like three agent tasks. But we are still in urgent need of models able to do longer and longer horizon planning. For instance, there is a new benchmark proposed by OpenAI, which is called PaperBench. They ask the models to do machine learning research on their own. And this kind of task needs more than 48 hours for even humans. So now, despite the 10-turn challenge has already been solved, we still have this kind of long horizon challenge, and we need more principled models to be able to achieve this. Finally, I will talk to you about my vision on the future of using agents to perform scientific discoveries. So one challenge is building comprehensive environments for scientific research. How to utilize currently available tools, softwares, and databases to build this kind of environments, because we know that tools are very expensive to build for even humans, and there is a lot of tools available there out there just for bio tasks. So here's an example, the Bio Mini paper. They integrate more than hundreds of available bio tools together. So they use human annotation to make all the tools in a single format, which is very easy for large language models to use. However, this kind of tool building requires a lot of human efforts. How can we make it more efficient? We propose a solution with our science board paper. So instead of creating a new tool for each function, we can use the wealth of softwares in scientific studies. So instead of creating a new tool, we just install all this kind of softwares onto a laptop environment. And then using multimodal models, we can ask the agent to do clicking and selecting, very similar to how we use the mouse on our computers. In this way, we can ask the agent to solve all these problems, to use all these softwares in a general manner. So in my vision, I want to build a comprehensive bio-agent system and to enable all kinds of agents. So first, I want to build an environment with various tools and softwares. MCP services, coding tools are just very similar like the Bio Mini paper, but I want to also incorporate all kinds of softwares which are very easy to incorporate at a large scale. Then I want to build a very flexible interface to interact with this kind of environments. I want to enable both coding interface so that model can use coding to execute all these kind of softwares and tools. I also want to build a SWE agent. So SWE agent can access the file systems and terminals and do things just like scientists do. And also GUI agents, as talked earlier, we can use like a user-friendly way to use all these kind of softwares. So now we have a good enough system. What kind of agents we need to build to understand this kind of science research? One thing we need to know is that in real experiments, the data are much more noisier and they're harder to perceive. In our database environment, we can still get some natural language text, which is very easy to understand for large language models. But this kind of experimental data are much more difficult. For example, here we have live cell imaging data, which is basically microscopy data, which is very commonly used, and also spatial transcriptomics data, which are many layers of data, including like pathology data and gene data, and organized in a spatial distribution. Also, humans are better at abstract reasoning than these models. For a large language model, it can easily give us a very simple description of those kind of experimental data. It cannot give us a very good conclusion. For instance, a human researcher could look at this kind of spatial transcriptomics data, which is basically things distributed in a spatial environment. So we want to know like what kind of interaction between those genes are happening out there. So a human will try to analyze this kind of data with maybe plots and with Python code, and they try to build some analysis to test their conclusions. And they also try to do some verification with knowledge. Maybe they try to Google it, they try to PubMed search some of their hypotheses to make sure the hypotheses are good. So how can we build agents to work in the same way as scientists to understand all those kind of complicated experimental data? For previous reason and planning tasks, we often use large-scale reinforcement learning to train this kind of models. We just give the large language model a reward based on maybe like rubrics, maybe some rules telling it like whether you're doing good or not. But this kind of systems are not good enough for training open-ended discovery tasks. For example, if you want to generate a hypothesis, it's very hard to test, and you cannot get a reward easily. So in my vision, I want to create open-ended RL systems that can work with agent as reward. So instead of directly giving the large language model a simple scalar reward, I want an agent to be able to verify its hypothesis in lab environments or virtual environments and use some tools to do some analysis And in the end, it can tell the model whether it's a good or not reward. Building this kind of systems not only require better algorithms, but also require improved infrastructure on doing async large language model training. So I'm sure like with efforts from both sides, both building better agents as well as building better environments, we will get more interesting scientific discoveries in the future. So thank you all for the talk, and here's the list of my collaborators, and thanks to my mentors. And I will be happy to take any questions.
Oh, thank you for the talk. We have a bunch of time, so please, anyone on Google Meet, so and plus time, feel free to ask questions.
Can we hear them? Oh, yeah.
I think so. We'll see if they're on you. Any questions?
Can I ask some political questions about your dense embedding model for the first part? So like, how did you train the model? Like, what kind of data did you use, and like what the piece model?
So basically, it's very similar to a BERT model. We have a large scale database like Unicode, and we take all the protein sequence here, and we do mask language modeling to predict the token that is masked. In this way, we can train very similar to protein mutation prediction. And then we will have a protein language model, and then we get a dense embedding the same way as we get a dense embedding.
Oh, so instead of training model directly using like contrastive training objective
Yeah.
dense embedding model, you like train like mass lens
Yeah.
model and use embedding.
I think it's not better. We also try like the contrastive learning. I think both works,
Mhm.
but I think like the easiest way we try to directly train with MLM is just
That's surprising.
Yeah.
So you wound up training your own protein language model, not just using like ESM2 or something like that.
we train both. So we try to do a scaling law analysis using our own model, and I think it's very similar to the results of ES1B. So both works. I think there are many off-shelf protein prediction language models that you can use very easily.
Oh, so you trained from scratch. I thought you just used the ESM model.
Yeah, we also train from scratch. But in the end, we just put the results of ES1B on the paper because it looks better.
I guess I have a question then, because I think it's been a while since I read the paper, but I think most of the sort of predictive protein language models that work well do use the multiple sequence alignment embeddings as part of their input. So if you're using that model that uses MSA sort of implicitly to get away from multiple sequence alignments, do you feel like there's any sort of like bias in there or concerns about that, or any benefit in just training something from scratch that's MSA free to get like a less potentially biased embedding space?
I feel like one of the reasons to use MSA is told in the ESM4 paper. So they do a scaling law analysis. They show that if you don't use MSA as input, you can easily get saturated when you do a scaling law experiment. So MSA is basically you need to understand the evolutionary information from a large scale of a large profile of sequences. And doing direct protein language model pre-training, you won't be able to understand the same depths as good as the MSA models. So it's like doing a faster scaling law with MSA as input. But I think like it's still a biased way because people just like to stick with some traditional methods in bio domain. So I think MSA is definitely useful, but we should not still use this kind of slow to make features in training. I think the retrieval is just working as well. So we can easily do some kind of retrieval augmentation for training on protein language models. I'm sure that somebody will test it out later, but there's no papers yet.
I have a question on the future work. So what there's the first part about building the tools and environments, which makes a lot of sense. And in some sense that's like all these tools exist. We just gotta
Yeah,
to make them accessible either through common formats or GUI agents and so on. What is there any, I guess, sort of technical challenges or like research challenges on that side? Or do you think it's a matter of just sort of sitting down and, cataloging all the software possible and testing it out?
yeah, I think we have all the components already, but one problem is like current systems are built independently. We have code agent environments, we have GUI agent environments, sweet bench environments. So people are not stitching it up. But I think like for using computers, it's okay to don't stitch it up because we have good softwares. But for bio, we definitely need to stitch it up because I think like for some tasks you have good softwares, but sometimes you have good headless mode softwares. So you need to do a better combination. So I think like it's very easy to implement. So basically it's just like putting every environment together and write a good Docker or virtual machine stuff. But I think it will be very valuable to write it, even though it's quite easy.
Separate question for the bio-based work. Do you have a sense of how much of the questions you tested were already in the literature that the models were trained on? Like to what extent is it, testing their memorization skills versus testing their ability to reason?
So we actually have a few rounds of filtering to figure out this kind of problem because at first when we create the database, there's barely any difference between the natural questions as well as the perturbed question results. And we found out it's because of memorization. So to make it better, so we first remove some very high citation papers. So we try to get the paper to be high quality, but not the most high quality papers, which the model will definitely memorize. And then we also remove the like the older words. We try to keep up with the latest words. So this can all help it a bit. So in the end, we can see that we have some questions that are harder to understand for models because it's novel.
But all the perturbations that you tested were experimentally verified. You're not
Yes.
like, just reasoning from the CAG pathway or something and saying, oh, probably this happens.
Yeah, we are worried about the quality of those kind of reasoning because it can be useful for training,
Right.
but it won't be so good for doing evaluation. So we still want our evaluation to be very accurate.
Sense. Yeah, I wonder if there's a big difference if you do a time cutoff or something.
Yeah.
Any other questions? Anyone on the call?